Yeah, so of course there are situations where sort of doing this type of hard clustering, finding discrete sets or categories where your cells should fit is not really the most appropriate approach. So in that case, we need to have a different approach, which is called pseudotime analysis. So in pseudotime analysis is sort of typically used when we have some sort of continuum. Most often we think of that as when we have some sort of differentiation process. So when cells sort of start out in one cell type or cell state, and they sort of gradually move or progress into a different stage over time. So the problem with the single-cell RNA-seq assay is that we can only measure each cell once. So because we lyse them and then they can't go on. So what will happen is that we essentially get like a snapshot of what's going on in our sample. So and if we have this type of differentiation process, what will happen is that some of the cells that we profile will be sort of at the earliest stages of this differentiation, and some of them will be closer to the end. But a priori, we don't know where each cell is. So really the goal behind Pseudotime is to sort of try to sort of align, essentially project or align the cells in a one-dimensional setting. And then we can sort of, once we've ordered them, then we can look at sort of different properties, such as expression profiles of certain genes, see how they vary, and sort of learn different things about this differentiation process. Doing this is sort of in the one-dimensional setting is sort of relatively straightforward. The much more interesting and of course more complicated situation occurs when you have branching going on. So this happens very frequently when you have two or more different cell types that sort of share a progenitor during development. So I'm showing you here on the left, arguably the most sort of well-studied and famous example, which is the hematopoietic system, where the hematopoietic stem cells will differentiate into various types of these blood cells. So what we need to do, again, now from a sort of mathematical point of view, is to sort of identify, ideally we would like to have an algorithm that sort of automatically identifies these branches, their endpoints, sort of both and their locations, and how many there should be. And this is, of course, much more challenging than doing just ordering them along a single dimension. So there are multiple methods for doing this. A few years ago, at least, there was a review, and I think it turned out that this is pretty much the most popular field within or area within the computational single-cell RNA-seq in terms of the number of algorithms that have been published. So the first method to do this was it's called Monocle from Cole Trapnell's lab. This is old these days. It's not really used, but there's Monocle 2, which is quite popular. So they use a reverse graph embedding strategy. And I mean, very loosely speaking, what they're doing is that they're essentially trying to find a sort of principal graph, which is a sort of extension of the idea of a principal curve, which sort of tries to capture as many of the points as possible by sort of going through their centroids. So Monocle 2 will sort of also automatically try to identify a number of branching points and their locations. So one drawback of not just Monocle 2, but most of these strategies or methods for doing this is that they can't really tell you that they're not directed. So they can't tell you where the process starts, and which is the starting point and which is the end point. In my opinion, this is not really a major drawback, because typically we can sort of have quite a lot of biological knowledge what's going on, and by looking at the profiles of the cells near the different ends, we can usually figure out quite easily which one is the start and which one is the end, or which ones are the end if you have branching. So a different method is called diffusion pseudotime. This is based on the idea of these diffusion maps, which is basically trying to find a sort of a low-dimensional diffusion kernel to fit the data. And it's sort of, you can think of it as it sort of tries to, based on the ideas of random walks, it tries to find the sort of the shortest random walks for connecting these different points. And again, this is a very robust method, which seems to perform very well. One of the these pseudotime methods, they really only work for doing sort of one-dimensional or sort of lower dimensional when you have a few branching points, but they don't really apply to sort of the type of big clustering studies. And if you want to understand how the cells are sort of moving or transitioning in that space, then it gets much more complicated. But fortunately, there's a very clever method called RNA velocity, which really effectively allows you to understand the sort of the direction of each of these points or the cells in this cell state space. So the idea is sort of based on a really clever observation that even though these different assays that we have are sort of, they look at polyadenylated RNA, you will still get some reads from pre-spliced RNAs. So you'll get some reads that correspond to introns and are hence unspliced. And then what you can do is that you can sort of set up this very simple first order differential equation for how the RNA gets transcribed and spliced, and then using this information about this number of spliced and unspliced reads, you can fit the parameters for this ODE, and then given these different parameters, we can infer sort of what is the temporal dynamics of these different genes, and then what we can moreover do is that we can look at the individual cells, and we can sort of place them in this phase diagram, and we can sort of infer for each cell if it seems to be in the process of sort of producing more transcripts, or if it's in the process of where degradation is dominating over transcription, so it's sort of going towards zero, or if it sort of seems to be close to equilibrium. So we can do this for each gene in each cell, and then we can basically calculate a sort of a velocity vector that we can project onto the same space, and that really allows us to to understand sort of how each cell is sort of moving around in this high-dimensional space. So this is really elegant and is really powerful for understanding sort of what kind of transitions you can infer from your data. So what's also really useful is that you can do even better. You can actually go back into and connect this to real time. So this was done, for example, in this single cell malaria atlas that I told you about. So there we have this life cycle of the malaria parasite, which is as the name implies, it's a cycle. So it makes sense to sort of plot the data in this type of cylindrical fashion. And then by relating this to other external data sets where people have taken sort of bulk sets of these parasites and sort of synchronize them and then profile them at different times, you can sort of identify certain key events, and then you can relate those to real times, and this allows you to understand how this snapshot type of data applies or correlates with the real time events. So I will stop here before we go on to the next. And